In 2023, a team of geneticists quietly published findings that almost no one outside of academic circles paid attention to. They had spent years collecting and cross-referencing DNA samples drawn from thousands of individuals across Canada and the British Isles. And when the results came back, the lead researcher on the project reportedly sat with the data for a long time before saying a word to anyone on the team. What the samples revealed was not what anyone had predicted going in. The patterns embedded in the Canadian genome did not line up the way the history books said they should. The British ancestry that Canadians had long assumed was the dominant and defining thread in their genetic story turned out to be only one part of a far more complicated picture, and the part that was missing pointed somewhere else entirely. The deeper the scientists looked, the more they realized that what Canada believed about itself, genetically and historically, was built on a foundation that had never been properly examined. This is what they found. If you want the unfiltered version of stories like this one, the full breakdown with additional sources and commentary is available on the Patreon. The link is in the description below. Now here is the full story. To understand why the results shocked the research team, you first have to understand what they were expecting to find, and why those expectations were so deeply rooted in how Canada tells its own story. Canada was, for most of its official history, presented as a British country, not merely influenced by Britain, but fundamentally British in character, in law, in culture, and in blood. The Dominion of Canada, established in 1867, was built on a constitutional framework modeled after Westminster. Its legal system was English common law. Its monarchy was the British Crown. Its largest cities named their streets after English kings and Scottish explorers. For generations, English-speaking Canadians grew up understanding themselves as a transplanted piece of the British world, connected to the mother country by something they believed was deeper than geography. That belief was ancestry. The assumption was not entirely invented. Britain did send enormous numbers of settlers to Canada across the 18th and 19th centuries. The waves of migration that followed the American Revolution, the post-Napoleonic economic collapse, the Irish famine of the 1840s, and the aggressive land promotion campaigns of the late Victorian era pushed hundreds of thousands of British Isles emigrants onto ships bound for Halifax, Quebec City, and St. John. By the time of the 1871 census, approximately 60% of the population of Ontario identified as having origins in the British Isles. In the maritime provinces, the figure was even higher in some counties. British descent was not a myth. It was a demographic reality, at least on paper. But paper, as the geneticists discovered, lies in ways that DNA does not. The study that would eventually produce the controversial results began not as an investigation into Canadian identity at all. It started as a comparative population genetics project aimed at mapping the regional diversity within the British Isles themselves. Researchers at institutions in Canada, the United Kingdom, and Ireland had noticed something accumulating in the literature for years without anyone drawing it into a coherent picture. The British Isles, despite being a relatively compact archipelago, contain an extraordinary amount of genetic variation. People from Cornwall do not look genetically like people from Orkney. People from County Clare carry markers that diverge significantly from people born 50 miles away in County Tipperary. The British Isles are, genetically speaking, not one population but many, layered by successive waves of migration from the post-Ice Age hunter-gatherers to the Neolithic farmers, the Bronze Age Beaker people, the Celts, the Romans, the Anglo-Saxons, the Vikings, and the Normans. If you were going to compare Canadian DNA to British DNA, you first had to decide which British DNA you meant. That turned out to be one of the first major revelations of the project. The concept of a single, unified British genetic profile is almost entirely a political and cultural construct rather than a biological one. When researchers began pulling together existing data sets from the UK Biobank, from Irish Biobank projects, from Scottish genetic registers, and from Welsh population studies, the internal variation they found was striking. A cluster analysis of British Isles DNA produced not one population blob on a genetic map, but something closer to a mosaic of distinct regional pools, each with its own characteristic distribution of haplogroups, its own admixture ratios, and its own echoes of historical migration events that official history had either minimized or forgotten. The English Midlands, for example, showed a significantly stronger signal of Anglo-Saxon ancestry than did the southwest of England, where older pre-Roman lineages had persisted with remarkable stubbornness. The Scottish Highlands carried genetic signatures more closely related to Irish populations than to the Scottish lowlands. Northern Ireland was genetically heterogeneous in ways that reflected its layered colonial and pre-colonial history. Wales was, in some of its inland valleys, the closest living genetic approximation to the pre-Roman Britons that the Iron Age would have produced. All of this mattered enormously for the Canadian comparison, because when Canadians said they were of British descent, they were collapsing this entire mosaic into a single label. They were not saying they were Anglo-Saxon or Scots-Gaelic or Cornish or Ulster Scots or Orcadian. They were saying British, as if that word described a coherent genetic package that could be found in a blood sample and matched against a reference. It could not. And when the researchers began comparing Canadian samples against the full internal diversity of the British Isles, rather than against some average composite, the mismatches started appearing almost immediately. The Canadian samples were assembled through genealogical societies, university research programs, and provincial health data partnerships with careful representation from Atlantic Canada, Quebec, Ontario, the Prairies, and British Columbia. The researchers also recruited participants who self-identified as having exclusively British ancestry going back at least 3 generations, attempting to isolate what might be thought of as the British-descended Canadian population. This self-identification, it turned out, was where some of the most interesting problems emerged. When the DNA of these self-identified British-descended Canadians was plotted against the British Isles reference populations, a significant proportion of them did not cluster where expected. They did not fall neatly into English or Scottish or Irish pockets of the genetic map. They fell in between, or clustered in unexpected directions, or showed admixture signals that pulled their genetic center of gravity toward populations that had nothing to do with Britain. The researchers initially suspected data error or sample contamination. They ran the analysis again. The anomalies persisted. The first explanation that the team seriously investigated was the French factor. Canada's French-speaking population has always existed in a parallel history to the English-speaking majority, and their genetics are, in certain respects, among the best documented of any population in the world. The Francophone Canadians who settled Quebec in the 17th century came almost entirely from a small number of French regions, primarily Normandy, Perche, Poitou, and the Île-de-France, bringing a genetic profile that was distinct from the British Isles populations in measurable ways. More importantly, they founded a community that was remarkably endogamous for its first century and a half, marrying within their group at rates that produced a pronounced founder effect, where the genetic variation of the entire future population was shaped by the relatively small founding group. The Quebec Biobank, one of the most complete population genetic databases in the world, has allowed researchers to trace Francophone lineages with unusual precision. And what they found is that the French genetic signal does not stay neatly confined to Quebec. It bleeds across the linguistic border in ways that parish records and census data do not capture. Intermarriage between English and French communities was officially rare during the periods when the two groups were most hostile to one another. But DNA does not record official attitudes. It records what people actually did. And what the data suggests is that across several generations of shared geography, trading relationships, frontier proximity, and the particular intimacy that isolated rural communities produce regardless of language, the genetic boundary between English Canada and French Canada was far more permeable than the political boundary. A person who identified as English in an Ontario County census in 1880 might carry French Acadian or Quebec Francophone genetic contributions from 2 or 3 generations back that no document would ever reveal. Because the family had anglicized its name, converted to Protestantism, and quietly merged into the English-speaking community. The DNA remembered what the paperwork forgot. The Acadian population added a further complication. The Acadians were French settlers of what is now Nova Scotia, New Brunswick, and Prince Edward Island, and their history includes one of the most traumatic episodes of the colonial era, the deportation of 1755, in which British authorities forcibly expelled approximately 10,000 Acadians, scattering them to the American colonies, France, the Caribbean, and eventually Louisiana, where their descendants became the Cajuns. A portion evaded deportation, hiding in the forests and marshes of the Maritimes, and others gradually returned afterward. Their genetic descendants are distributed across Atlantic Canada in numbers not fully captured by census data, partly because many families in the 19th century found it safer to present themselves as simply Catholic rather than specifically Acadian. When maritime Canadian samples were subjected to ancestry deconvolution analysis, Acadian genetic contributions surfaced in populations with no Acadian surnames and no family oral tradition connecting them to French settlement. But the French factor, while significant, was not the result that silenced the room when the research team assembled to discuss their findings. The result that produced genuine shock was something that none of the researchers had built into their initial hypothesis. It was the Indigenous signal. Canada's Indigenous peoples, the First Nations, the Métis, and the Inuit, have their own distinct and ancient genetic heritage, descended from populations that crossed into North America from Northeast Asia across the Bering Land Bridge, somewhere between 15,000 and 25,000 years ago, and then diversified across the continent over millennia into hundreds of distinct nations and language families. Their DNA carries genetic signatures, including certain haplogroups in both the mitochondrial and Y chromosome lines, that are not found in European populations and that serve as clear markers when they appear in an admixture analysis. The researchers were not naively expecting to find no Indigenous contribution in their Canadian samples. The existence of the Métis people, a recognized population of mixed Indigenous and European ancestry, had been part of Canadian history since the fur trade era. What they were not expecting was how widely distributed the Indigenous genetic signal would be across samples that had no Métis identity, no Indigenous community connection, and no family awareness of Indigenous ancestry whatsoever. The signal was not enormous in most individuals, but it was present at a frequency the team's statisticians found highly unlikely to be explained by chance or data error. It appeared across regional samples in families with deep English Protestant genealogical records and in communities that had historically positioned themselves as the most thoroughly British of all settlement groups, including United Empire Loyalist families who had maintained, across generations, an almost mythological pride in their purely British bloodlines. The researchers were careful in how they interpreted this. The presence of Indigenous genetic markers does not necessarily mean recent intermarriage, though in some cases it almost certainly does. It could reflect admixture that occurred very early in the colonial period, in the 17th or early 18th century, when the fur trade created sustained cross-cultural relationships, and when the children of those relationships sometimes entered European-identified communities under European surnames, without any record that later genealogical research would capture. In other cases, the signal likely reflects admixture through Métis intermediaries who married into communities that did not acknowledge that background. What the finding meant, in practical terms, was that a measurable portion of Canadians who had spent their entire lives believing themselves to be of purely British descent were carrying genetic contributions from the peoples who had lived on the land before any European ship arrived. That history had been erased through assimilation and name-changing and community forgetting. But it had not been erased from the genome. The researchers described it as among the most striking examples they had encountered of the gap between documented ancestry and biological ancestry. The documented ancestry said British. The biology said something more complicated. The Scottish dimension of the study produced its own surprises. Scots were among the most numerous immigrant groups to Canada, arriving in waves from the Highlands and Islands, the Lowlands, and from Ulster. But Scottish genetics are themselves a layered inheritance. The Highland Scots cleared from their ancestral lands after Culloden in 1746, and again during the agricultural enclosures of the late 18th century, carried genetic profiles that diverge significantly from lowland Scots. Highland genetics show stronger affinities with the Gaelic-speaking communities of the west of Ireland, reflecting the shared Gaelic world that once stretched across the northern Irish Sea. When Canadian samples from communities with known Highland settlement history, places like Cape Breton Island, were plotted against the British Isles reference map, they clustered closer to the west of Ireland than to the English Midlands, even though both would conventionally be labeled British. The Ulster Scots added yet another layer. Transplanted from Scotland to Northern Ireland in the 17th century, then migrated again to the American colonies, and eventually to Canada after the Revolution, their DNA is a palimpsest of displacement. It carries Scottish genetic base signatures overlaid with Irish admixture from their Ulster decades, sometimes further mixed with English Protestant settlers under the same plantation scheme. When Ulster Scot samples were analyzed, they occupied a genetic middle space that required the full diversity of the British Isles dataset to properly describe and did not match cleanly against any single reference population within it. The Loyalist communities of Ontario and New Brunswick added another complication that proved more significant than anticipated. The United Empire Loyalists were not, as a group, exclusively of British descent. The American colonies from which they came had been settled by people from across Northern Europe, and by the Revolution, the colonial population included substantial numbers of German-speaking settlers, Dutch families from the Hudson Valley, and Huguenot French Protestants. When these families came to Canada, they brought British political allegiance, but not always British genetic ancestry. A Loyalist family with a name like Snyder or Van Camp was carrying German or Dutch ancestry that plotted comfortably on a Rhine Valley reference map rather than an English one. The researchers also identified a signal that they had not anticipated: a Norse or Scandinavian contribution, particularly visible in samples from Newfoundland and coastal Labrador. Newfoundland's documented history of European contact begins with Cabot in 1497, but the Norse were at L'Anse aux Meadows 5 centuries earlier, and the DNA suggested their demographic footprint on the region was greater than the archaeological record had confirmed. The Norse genetic signal exceeded what later Scandinavian immigration could explain, and the most plausible interpretation was sustained contact with indigenous populations across a period longer than the single archaeological settlement layer implied. The prairie provinces produced a different genetic picture entirely. The homesteading era brought enormous numbers of settlers from across Eastern and Central Europe: Ukrainians, Poles, Czechs, Germans, Hungarians, and Scandinavians, who were explicitly non-British and knew it. But the degree of intermarriage between these Central European communities and the British-descended prairie population was substantially higher than census data had suggested. By the third and fourth generation, individuals who identified simply as Canadian often carried admixture signals pointing toward Ukrainian or German or Scandinavian ancestry at the 5 to 20% range, alongside what they had understood to be exclusively British or Irish family lines. The researchers also found, particularly in British Columbia, a signal that reflected the province's Pacific history. Chinese immigration in the 19th century, driven by the gold rushes and the construction of the Canadian Pacific Railway, was followed by anti-Chinese legislation designed in part to prevent demographic mixing. The study found that mixing had occurred anyway, quietly, outside what polite society acknowledged. The East Asian genetic contribution in certain British Columbia samples was present in people who carried no family knowledge of it, absorbed into family histories that had left no record and that predated living memory by several generations. When the full picture was assembled, the research team found themselves looking at a genetic portrait of Canada more diverse and more deeply mixed than any official narrative had described. The Canadians who thought of themselves as British turned out to be, on average, somewhere between 60 and 75% British Isles in their genetic ancestry, with the remainder drawn from French, Indigenous, Central European, Scandinavian, and other contributions that varied by region and family. That range sounds high, but in the context of a population that had been telling itself for 150 years that it was essentially and overwhelmingly British in blood, it represented a significant discrepancy. In some of the communities most emphatic about their British heritage, the British component was lower than the regional average. The comparison with British DNA produced its own set of revelations. When the Canadian samples were laid against the full diversity of the British Isles, the Canadian sample was, in some respects, more genetically diverse than any single British region, because it had drawn from multiple British regions simultaneously, while also absorbing the non-British contributions described above. A Canadian whose ancestors came from multiple British counties, who also carried Acadian French, and whose family tree included a Métis great-grandmother absorbed into the English-speaking community, was not a diluted version of a British person. They were a distinct genetic entity, assembled on Canadian soil from components that had never been combined in quite that way before. This suggested that Canada had, over its relatively short history, genuinely produced a new population rather than simply transplanting the old one, and that the non-British components folded into the Canadian genome across 4 centuries of contact were equally real, equally formative, and far more present than the history books had acknowledged. The publication of these findings generated a reaction that split along cultural rather than scientific lines. Among population biologists, the study was received as solid and methodologically sound. Among heritage organizations, particularly those focused on loyalist and British lineages, the response was more turbulent. Several questioned the recruitment methodology for the self-identified British ancestry group, arguing the sample had not been carefully enough screened. The researchers maintained that the patterns were robust enough across sample sizes to survive that criticism. Some commentators used the study to advance arguments about multiculturalism and national identity that the researchers were careful to say the data did not support in either direction. DNA is a record of biological ancestry. It does not tell you who belongs or who does not, who is more or less authentically Canadian, or what any policy should be. It tells you who mated with whom across generations and what the full biological history of a population actually was, as opposed to what that population had been told. What the study did do, in a way that the researchers found genuinely valuable, was complicate the simplistic story. Canada had been sold to itself and to the world as a country with a clear and knowable British core. That story was not a lie, but it was a simplification so dramatic that it functioned as a distortion. The British contribution to Canadian genetics was real, was large, and was primary in the sense that British colonial institutions and British settler communities had been the dominant social and political force in shaping the country. But the genetic record showed that Canadian biology had never respected the borders of that story. It had always been mixing, absorbing, incorporating, and evolving in directions that the official narrative had no room for, because those directions pointed toward Indigenous peoples who were supposed to be separate, toward French communities who were supposed to be apart, toward immigrant populations who were supposed to maintain their distinctness, and toward ancestors whose existence in the family tree had been deliberately forgotten or quietly erased by the people who came after them. The scientists concluded their published paper with a statement careful and measured in the academic way, but carrying an emotional weight that anyone who spent time with the full dataset could feel. They wrote that the genetic history of Canada, like the genetic history of most settler colonial nations, is more complex and more humanly diverse than official accounts have generally acknowledged, and that the patterns visible in the contemporary Canadian genome represent not a problem to be resolved, but a history to be understood. The people in those samples had not chosen who their ancestors were. They had inherited whatever was passed down across centuries of human encounter. Whether that encounter was loving or coerced, recorded or erased. What the DNA contained was the full truth of those encounters, stripped of the narratives that later generations had built over them. For many of the Canadians who had participated in the study, the personal experience of seeing the numbers was not what they had expected. Some found the results affirming, confirmation of family stories passed down but never proven. A woman from Cape Breton, whose grandmother had always whispered that there was some Indian in the family, saw a number in her results that confirmed what the grandmother had known. Others filed the numbers away and went back to their lives. A man from a Loyalist family in Ontario, whose relatives had always emphasized the purity of their British heritage, saw a composition that included Indigenous and Central European contributions, alongside the expected English and Irish components, and spent months trying to reconcile what the data said with the story he had been raised inside. The deeper significance of the study was what it said about the relationship between identity and biology in a country that has spent its entire modern existence trying to define itself. Canada has always struggled with the question of what it fundamentally is, and the answer that official history provided, that it was essentially British with a French province attached, was never the complete answer. The DNA study did not replace that incomplete answer with a clean one. It confirmed that the question is more complicated than the official version allowed, and that the genome of the Canadian people contains evidence of a history richer, stranger, and more fully human than any simplified account could accommodate. The scientists who did the work were trying to read a biological record accurately. What they found was that the record, read accurately, tells a story the country has not yet finished learning how to hear.